I wanted to like make this general-ish because I'm just talking about in general how to find, access, reuse open data sets. So I'm not going to go too much into details of some of these tools, but if you want me to, I can. Like I can just start talking about anything. So just if there are specific things that you want to know about, or specific tools that you want to know about, things that you know that I have used, or wondering if I have used, or whatever, or how I got this data set or that data set, or I will be happy to tell you. Okay, so I would just start with letting you know why I'm here. Pretty much last year I won this Open Science Award, and then I think that really kicked off actually the Neuro Urban Helga Cooper Foundation Open Science Award. It's like a neuro thing, so you can apply next year. Anyways, I said the whole name. Like it. Anyways, so I won this award last year, and I think that like kicked off. People kind of associated me with open science because I won an award, right? And then they were like, okay, so she's probably an expert in open science, so we're going to invite her to talk about open science stuff. And that happened like for the Unique Student Symposium. It's happening here. It's happened other times. But what they don't know is that like the whole award thing was a bit of an accident. So I like won that award, but I didn't start doing open science because I was like, open science is a great thing and we should all do it. And I really want to do it. And this is how I'm going to do it. It was like I was doing my PhD and it kind of had a lot of open science themes sort of by accident. Actually, honestly, it was mostly because this guy, Ross, who's going to come up again in my presentation, but like Ross Markello, he used to be a PhD student in the lab. He like has graduated. He's now making a lot of money in the Silicon Valley. But like he was in the lab and I really admired him and he was doing open science stuff. And I was like, Ross is cool, be like Ross. So I was doing open science stuff too, kind of just I wasn't thinking about it. I didn't really know what it did or why it was a good idea. And then it was really integrated in my PhD from the beginning, and that ended up in me being awarded with this Open Science Award and people asking me to talk about open science. And now I'm like, well, maybe I should like know what open science is then, which has actually been really helpful because now I've learned a lot about open science. But so I guess what I want to emphasize is that like I'm still early in this open science journey, and really importantly, like this is so obvious, but I don't think people internalize it, that you don't need to be an expert to get started. So a lot of people feel like you need to know all the open science stuff and like come to lectures about how to access big data sets and whatever. In my case, a so-called open science expert, I like was thrown in the deep end. I had no idea what I was doing, and somehow I managed. And so you can too, you know what I mean? Like you can just get started, and I believe it'll work out. Okay, so what am I going to talk about? Because like finding open data and like accessing open data and using open data and big data in general, it's like very personal to you and to the data set. So I can't really give you this is how you're always going to find and use big data. But what I can tell you about is my personal experience, how I got there, and then hopefully that will be helpful. I'm going to be talking about a lot of general tips, which hopefully you can take away. But also, again, if there's anything in particular that you want me to focus on, if there's a method that you want me to go in more depth in, if you want me to start live coding so you can see all my typos, I will happily do that. So first part of how I used open data and integrated it into my work. So my work is very much multimodal, using different data sets, bridging them together into one PhD project. Second part is going to be how I found and used closed data and integrated that in my work, and then helped to make it open. So that's one thing that's maybe people don't know about, is that you don't just have to use open data. You could see that there was some data that somebody used in a paper and that you kind of want to use, and then you like ask them, can I use that data? And then maybe they say yes. And then that was like my whole PhD. And then I have this third part, which is actually just one slide, and the entire slide says credit where credit is due. So I just want to emphasize that like as a data leech, like it's really important to recognize the amount of time and effort and money and blood, sweat, and tears, and like the whole thing that goes into collecting these data sets. When we use them, is to give credit to the people who actually collected it and made it available. Okay, so part one, how I found and used open data in my work. So I'm just going to do this in a bit of like a narrative storytelling way. I'm going to start with like my very first project. So my PhD in general is sort of about bridging micro scale to macro scale. So if we imagine like the brain is this integrated organ, it works on many different levels of description, going from, let's say, gene transcription all the way to brain function. And now every point here, there are data sets out there. So for example, gene transcription on human brain atlas. I'm going to talk about this one in a bit more depth. Brainstem is another one. Abagen is a tool. Receptors, there is the NRU in Denmark. They're collecting receptors from receptor atlas. USpace coming out of Jülich in Germany. That's another collection of receptor profiles. I'm going to be talking about an atlas of receptor profiles that I put together later today. Synapses, there is actually data sets about like the mouse synaptome. This is just the tracer for you CBJ. They don't have the logo, so that's why. Yeah. The Big Brain Project. So we're here at the M&I, and I don't know if you guys know, but if you go up like 3 floors, there's a room with a massive TV, and Claude Lepage will like happily sit you down in a chair, give you a pair of 3D glasses, and like zoom through the big brain, which is this super finely reconstructed cellular atlas, or just in general anatomical atlas of the brain. And you can sit there with 3D glasses and see him like spin around the hippocampus or whatever. It's really fun. I highly recommend it. You're here in person, which means that you can go up 3 floors and do that, but maybe do it after my talk. Brain structure, and there's so many things, but for example, the Human Connectome Project, this is also function, but this could be like gray matter, cortical thickness stuff, diffusion weighted MRI for structural connectivity, and then brain function, which again can be a lot of stuff. Enigma, this is this big working group. They actually link a lot of gene expression stuff too, like GWAS studies and neuroimaging studies, but a lot of their work is looking at case versus control differences in cortical thickness. And specifically in disease. So that's why I have it under the function. It's kind of like dysfunction, but it could also be under structure and gene transcription. Neurosynth, I'm going to talk about in more detail during this talk, and the UK Biobank, that also could kind of be anywhere here. The point is there's a lot of data sets out there. And so the first step in finding and using them is a Google search. That's like the theme throughout this whole thing. The themes are Google and ask people for help. You have the spoilers, like that's what it is. Okay, so there's a lot out there. I'm going to focus on a couple, and in the context of my very, very first paper. So newbie Justine like joined a lab, and I had this kind of idea for a project. And I'm just going to, don't worry about the science, honestly, who cares about the science right now? But I'll just try to give you the gist of it, which is that we know that gene expression relates to proteins, relates to cell types, relates to synapses, relates to brain connectivity, eventually functional connectivity, eventually behavior, movement, cognition, whatever. And so if you like skip all those in between steps, what is the link between gene expression and brain function? That was the central question. And to do that, I wanted to do this mapping between gene expression on the one hand, brain function on the other hand. Okay, so what's the data? Because I have never collected data in my life. So I like needed data. The gene expression is coming from the Allen Human. Definitely big data. It's 6 people. So in that sense, it's not really big data, but it's like 20,000 genes. So again, big data kind of depends on which dimension you're talking about. I'm going to go in detail about, has anybody here used it, the Allen Human Brain Atlas? Nice, 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 nice. Does anybody here not use it and care about how to use it? Ish. Okay, cool. Okay, so I will talk about it in some. I can talk about it in more or less up to you guys. So the Allen Human Brain Atlas, what they have are 6 brains. 2 of them have both hemispheres. 1 of them just has 1 hemisphere. Okay, that's kind of important to remember because it means that there's 6 left hemispheres and 2 right hemispheres. It's not a full, it's not 6 full brains. They do an MRI to get the anatomy down. They're going to slice up this brain. They're going to image. This is these dead brains, by the way, guys, like people who are dead. They're going to slice up the brain. They're going to image it, Nissl staining. They section it. Then a couple things. One is like delineating the anatomy of these sections. The second thing is pretty much throwing a bunch of like fluorescent stuff, right? So that you can later label the genome, the gene expression of these different genes that are being expressed in these different cell sections. Expert structural annotation. So then eventually what you get is you get to the microarray. For those of you who don't know, microarray is this thing. Pretty much at every one of these circles, you're going to have some probes on a probe. Like the lingo is not great, but what the probes are is like little RNA or big half DNA, and it's wiggling there. And they're all of the same series, so that when you have your mRNA that's been transcribed from your brain section from these brains that you just sectioned, and they're glowing, when they bind, it's going to make that patch glow more. So can you see my mouse? Yes, you can. So here, for example, this would be for a very specific series of base pairs, in other words, a gene. And if it glows a lot, it means that in that section of the brain, there was a lot of gene expression or a lot of mRNA present for that gene. Whereas this guy over here, it's not glowing very much. So that means there was much less mRNA for that gene. So there's less gene expression for that gene. Got it? Nice. And what that's going to tell you is, well, okay, so they do the microRNA, then they match their section brains to the the MRI, so that they have the anatomy. How do you store the data? That's a whole other thing. That could be its own lecture. And then bingo bango, here's the Allen Human Brain Atlas. This is the thing that I used. Okay, so what is it exactly? Now, before I get into that, I just want to say, like, my really quick way of explaining the Allen Human Brain Atlas, you might have noticed, one of the struggles of integrating a lot of open data sets and big data and like a million different data sources and techniques, right? This is like microarray. Like, what is microarray to somebody who's doing neuroimaging every day? And what is like I don't know, receptor binding stuff to somebody who's doing like cognitive neuroscience, right? Like one of the struggles is that you're never going to be an expert in every single method that you're using, because you can't be. Like the real experts are the people who spent years like doing this and know it like by heart. I'm here trying to tell you what the Allen Human Brain Atlas is, but obviously if you get somebody from the Allen Human Brain Atlas, they're going to be able to explain it much better. They're going to know it much better. So it's just the caveat. It is like a challenge. I think one of the bigger challenges of doing this kind of multimodal work. Okay, so at the end, what you're going to get is you get this, for all the different brain regions, you have a gene expression level, okay? Brain regions by gene expressions, that's data, that's a matrix. You could store it in the CSV. Now you thought I was done talking about the Allen Human Brain Atlas, there's still a bunch more, which is all the processing. So that's just what, this is what the Allen Human Brain Atlas did. And then after that, people were like, okay, how do we use this in a way that's meaningful? There's a bunch of processing to be done. I'm not going to go too much in detail because, well, depending on people, I don't want to bore you guys pretty much, but you need to like select which probe matches which gene. How do I associate my brain section from those donor brains to an actual ROI that I'm interested in? How am I going to account for individual variants across these 6 brains? Am I going to filter out the genes that are really variable across the 6 brains? Am I only going to keep those that are very stable, or how am I going to quantify that variability? Yeah, this is the gene filtering. Eventually you get this region by gene matrix, and then you also have to be aware that there's this thing called spatial autocorrelation in the brain. I'm not going to talk about this too much. Some of you guys already know. But essentially we expect that gene expression will be similar for brain regions that are close to one another. So we have to take that into account when we do statistical methods. I can talk about that more if you want me to, but I won't. I think probably not the crowd. Okay, so there was the whole data thing, accessing that. And then there's like a million processing steps that it's like, oh my gosh, let's say that you're a cognitive neuroscientist and like you barely ever think about gene expression, and now you're being thrown with like all this information, which is very overwhelming. And then, by the way, here's just a list of all the different options you can like decide on how you want to get your gene expression. Like it's really overwhelming. A lot of open data science is really about using open data is about exercising these data science skills. And what I mean by that, honestly, is not necessarily that you have to reinvent the wheel every time you work with a data set. There are tools that are there to help you. So if something is overwhelming, if you have like come across a big data set that you think you want to use, but maybe you're not an expert in, you're not the only person. The person who put out that data wants it to be used. You know what I mean? Like, I mean, the selfish reason is like they want to get cited, but also they just like good for science. Like they put data out there, they want it to be used. There's probably a tool out there to help you. If there isn't, the person who put out the data will probably be willing to help you. Like, being an extrovert is so helpful because you can just send people emails and say, hello, please help. And the introverts are like, I would never do that. So then you have to like make an extrovert friend who will do those things for you. That's my advice. So in this case, for the Allen Human Brain Atlas, there's this toolbox called abagen. It's in Python. I'm sorry for the non-Pythoners. It's well documented. This was developed by Ross, by the way. Again, second time it comes up in this talk. This is the code. Import abagen. Gene expression equals, take my package abagen, like ABA Allen Brain Atlas genetics gen. That's why it's called that. Function is called get expression data, and then you give it the atlas you're interested in. Let's say you want it in Schaefer 400. Let's say you want it in Dutch-Kenyani. Let's say you don't want it in an atlas. You want the whole for every single sample that they did in the brain. And then this is going to spit out a brain region by gene expression for the 6 donors. If you want to go further, you can also take an average across those 6 donors, and you have your gene expression matrix. If you don't want all the genes, because you're like, I don't want to deal with this big data nonsense, you can also pick the gene of your interest. Let's say you were specifically interested in SNCA. Is that the one about Parkinson's? Anyways, whatever, like some gene that you're interested in, and then you can say, I just want the expression for that one gene, and you're going to input to get expression data. Here's the gene I want, and it'll give you the output. So it's going to seem a bit reductionist, but pretty much we went from like a big data source that like newbie me was supposed to use in my analysis for this scientific, I mean, bringing it back, right? I'm talking about this very first paper I ever did. The analysis I was trying to do, I needed data. I didn't collect it. I fetched it from here. And what it really came down to was figuring out that there was a tool I could use, asking a lot of people for help, like that was a big part of it. And then 1 line of code. Okay, so that's what this gives me, brain regions by gene. What was on the other side? So remember this whole question, the scientific question here was bridging gene expression to cognition. And on the cognitive side, I have this thing I'm going to call brain region or nodes. by terms. By terms, I mean, I say terms, I mean something like psychological processes. I'll explain that. Here I'm using something called Neurosynth. Who here has used Neurosynth? Okay, cool. A few people. Who here has heard of Neurosynth? Oh, okay, fair number of people. Who doesn't use it but might want to use it? Okay, cool. Okay, nice. Okay, so then I will explain what this is. So Neurosynth is this tool that I think came out in 2011, Tagliarconi et al., 2011 Nature Methods. And what it's creating is these meta-analytic associations between brain voxels and psychological processes, cognitive processes. Really what it is when I say psychological processes, it's a word. It's an association between a word and a voxel in papers. So it's asking if a paper or if a lot of papers have published on the word pain a lot, probably these papers are about pain. It's an automated meta-analytic tool, right? So nobody is going there and saying, is this paper actually about pain? Maybe it's about something totally different, but somebody saying pain all over. I don't know, you know, but like probably if the word pain is being mentioned a lot in the paper, or the papers are about pain, what are the coordinates, the voxel coordinates in the brain that they're mentioning? And then they're going to make this meta-analytic mapping between those brain coordinates and what that paper was about, or what those papers are about. Okay, so you get a probabilistic map. So let me go into a bit more detail. It's automated. It's across more than 14,000 papers. So that makes this very, very large scale. That's where the big data part comes in. And so I'm not going to, let's just pretend we have one study. What Neurosynth has done is that they have this bank of words, about 10,000 words. And for every paper, they're going to say what words show up at high frequency, like 1 every 1,000 words, I think is their definition of high frequency. So for example, if the paper is saying the word pain a lot, you would check off the pain box in your list of terms. At the same time, it's going to ask what voxels are being reported, because I don't know if you guys know, but in meta-analyses, especially like 10 years ago, it was very common to do your meta-analytic like fMRI thing. I did not mention until now, this is fMRI studies. I should have said that at the beginning. So in any kind of fMRI study, what they would do is they would say, we did an fMRI study of working memory. We found these significantly activated voxels. They would put it in a table, like actually XYZ coordinates in the table. And what's really great is that we are super, for the neuroimagers of us, we're super, super lucky that we have standards in the field. Lots of other fields do not have standards. And one of the standards we have is the MNI 152 template. And so what this means is that when people published, these are the areas in the brain that were activated in my study, those numbers could translate, right? Like you could, one person said an MNI 152, another person also said an MNI 152, they're saying the same language, instead of having to like point vaguely, is the part that lit up, which is much less specific. That is like I don't know if like for the neuroimagers of you guys, like be really grateful for the standards that we have. It's actually really awesome. It makes a lot of science easier and you don't even notice it. And it's something that I think a lot of the other fields are struggling with, trying to like share their data and advance like scientific output and cross collaboration when you don't have even like file type similar. Like how do I even like open this file? Please tell me, you know? So okay, enough of my soapboxing. Okay, so the idea here is you have a study, you're going to make a contingency table for a voxel and a specific term. Does that term show up at high frequency? Does this voxel show up in the table? For example, does pain show up a lot? Yes. Does voxel A show up? Yes. Or it could be something like, no, pain doesn't show up, voxel B does show up, or nothing shows up, reading doesn't, voxel B doesn't, whatever. For every couple of all these different terms, 10,000 terms or whatever, however many, and all the different voxels. Then when you do that across all these different studies, 14,000 plus studies, again, this is automated, this is not a human being doing this, what you end up with is a probability that a specific voxel is associated with a specific term across many different studies. And so the question is there a dependency between voxel activation and the presence of a term in the literature? There's two ways you can ask this question. You can say, what is the probability that a term is published or mentioned in the paper at high frequency given voxel activation? Or the flip side, what is the probability that the voxel is there in a table given the term shows up a lot at high frequency? Okay, so the kind of two flip sides. I usually use the association test. I think generally the recommendation is to use association test, but honestly, I'm not totally sure why. Okay, but you can see here, right? So it's like brain map of voxels. Given this brain map of activated brain regions that have been reported in a paper, what is the likelihood that this is related to working memory or emotion or pain or whatever? flip side would be, given the fact that my papers have the likelihood that I see this voxel activation. Okay, and at the end of the day, you're going to get this matrix of brain regions by psychological processes with association. Really, it's probability, but it's an association between specific brain region and voxel. Okay, so how do I use this tool? Like, how do you use Neurosynth? How do you access this data? You Google neurosynth.org. Like, honestly, that's what it is. And then you go to the download tab and like download some stuff. Or Neurosynth GitHub. I did this Halloween, that's why it's like still Halloween vibes. But like, I think what I did was I used their GitHub. So they have like a nice web app, you can use that. But I kind of used the GitHub tool that they have this sort of behind the scenes, which you can find by Googling Neurosynth GitHub. And like the real answer is PS Ross helped me. Like that's how that happens. So if you want to use Neurosynth, if you want to try to figure out on your own, yeah, like great learning experience. But if you want to ask somebody for help, like me, ask me, you know what I mean? Just send me an email, say, I would like to use Neurosynth, please help me out. It'll take some time, but I'm more than happy to sit you down. I didn't want to do it here just because some of you probably don't care at all. So I don't want to like bore you about it. But often when it comes to these tools, find a person who's done it before, ask them for help. They're probably friendly. Yeah.
I'll just add, I'll talk about more about this at the end, but Justine has some office hours available that you can sign up for on the Open Science Office Hours website. So literally, if you want to learn how to do this, you can sign up for an office hour this month and she can help.
Yes, that's true.
But I'll show you how to do that at the end.
Yeah, you can also ask me stuff that is not related to this. And then I will be honest if I do not know what you were talking about. And I'll say, I can't help you with that, sorry. But maybe I know who could help you with that. So yeah. Okay, so at this point, we have the gene expression. So remember, this is newbie Justine. She's joined the lab. She's trying to do this like project. I need gene expression data. So I'm like, oh my gosh, Island Human Brain Atlas. Ross, please help me. And like, that's pretty much how that came to be. Brain regions by genes of gene expression. On the other hand, I want this kind of measure of functional specialization of different brain regions. So for that, I'm going to use Neurosynth. And then you're like, well, how did you know that these data sets were things? And that's like a great question. I think the real answer of how I knew is that like the lab knew and they told me, but that's like a really boring answer. If I were you, I guess like, or when I'm trying to figure out what the new data sets are, there's definitely like an element of Google involved, but also like, I mean, hopefully you guys do this, but when you do like your regular reading of papers that you read to stay up with the literature, just keep an eye out for when people or like the different stuff that they use. If you thought something was cool and you're kind of interested in that data, just like I don't know, jot it down or like keep an ear out for that. You know what I mean? For like, okay, that's a data set that I could use. Should I contact them? Is it in their GitHub? So much data is in open GitHub repos. Like they might not tell you, but it's there. And I mean, you should let them know that you want to be a nice person, but like it's there. Like there's a lot of stuff that's out there. Okay, so at this point, I have these two big data sets. My matrices are like, I think this was 200 brain regions or so by, I filtered out some genes, so it was like 8,000 genes. And then for the terms, it was 200 brain regions by, I think it was 123 terms, or 123 terms or psychological processes that I was looking at. And I wanted to make associations between genes and terms, which is a little bit like I had a big matrix on one side, another big matrix on the other side. How do I map those together? This is another like example of using big open data. It's a lot of like these kind of data science skills, machine learning skills, organizing and cleaning data, like just the amount of time of just downloading the CSV, seeing that there are nans in the data. How do I deal with the nans? Am I going to interpolate? Do I ignore those? Do I remove those brain regions? You know, like this kind of relatively like simple in the sense that you don't necessarily need somebody to like show you how to do it, but it's going to take time. Sometimes it's a bit annoying. It's like persistence and patience. Like, honestly, that's what it is. I see nods. You guys know. You guys have like been through it and you guys know. In this example, I just like want to let you know what I used. I used something called partial least squares analysis. So what's nice about that is I didn't want to do these one-to-one correlations or mappings between every single gene and every single term. I wanted to do something multivariate that takes into account all of my genes, all of my terms. And so what partial least squares analysis does is it takes these two, multiplies them together, matrix multiplication. So if you imagine genes by, for those of you who remember your linear algebra, genes by brain regions times brain regions by terms is going to give you genes by terms, right? Because the middle dimension, you guys are like throwback to linear. Yeah. You're going to get this covariance matrix, and I'm going to apply singular value decomposition to that. What singular value decomposition is going to do is it's going to decompose my matrix. Sorry, am I losing you guys? I'm almost done. Singular value decomposition is going to decompose your matrix into USV. So those are three matrices. So U is a square matrix, S is a square matrix, V is also a square matrix. that true? No, U is not square. Wow, I should not be. U is genes by brain regions, V is brain regions by brain regions, and S is brain regions by brain regions, and V is brain regions by terms. Okay, anyways, we can talk about that office hours. So the point is, it's going to give you a weight for every gene, a weight for every term. That's the U matrix and the V matrix, and the S is the singular values. It's going to decompose your data. I'm so sorry that you guys are like seeing this. It's going to decompose all your data into a series of latent variables. Latent just means it was like always there, but you didn't see it. So it's not one of the variables like this is a gene. It's like, here's an axis of variation that these two are associated with one another along. And you just have to apply singular value decomposition, or also called PCA, by the way. Just these were first, and then that's PCA, to see this axis of variation. And then that's what this paper was. It was me looking at that first latent variable. It was the first one that was the only one that was significant, statistically significant. So that's just an example of one tool that I use to work with this big data. You can use other things, ML, that's big, artificial stuff, AI stuff, that's also big, like, you know, lots of things you can do. The one piece of advice that I would give like early me, or even me now, or anybody, is like really plot your data. Like, it's so easy to download these big matrices, import them into whatever Python, R, MATLAB, whatever it is you use, and then like run with it, you know, like run PLS analysis, get your output, write a paper. Like, no, you need to get a set. So when you get a matrix of data, if it's a matrix, plot it as a heat map. Just take a look at it. Because then you're going to see, if you have one, let's say, person or brain region or whatever the variable that's black the whole row down, you know something's wonky, and you never would have known that if you had just done your like analysis and ran with it, right? So please plot your data, look at it, take a second to think, do these associations that I'm finding make sense? What does it look on the brain? So, I mean, in my field, because it's more neuroimaging, I'm plotting brains all the time, and that's like a great sanity check. It's like, whenever I have something, plot it on the brain. Does it look right? Or is it like the same value everywhere and then negative 1,000 and one, you know, like these are the kind of things that you can find. Oh my God, scatter plots. Plot your scatter plots. Don't just like, here's a correlation coefficient. No, look at it. You know, do you believe it? Do you like honestly think, like, will people laugh at you if you say that that is a correlation? You know what I mean? Like, that's like what's going on in my mind. Are people going to laugh at me? Like, that's mostly what. So yeah, plot it so you know, so that you can feel confident when you tell people, yes, it's the correlation of 0.3. I know it's just 0.3, but I believe in it. You know what I mean? I mean, I guess that also like it's really beneficial for optimists, just like believe in everything. But anyways, okay. So I've talked about two big data sources, about how I came to use them. I, as a not big data expert, especially not 3 years ago, right? Like 4 years ago, whatever it was. I didn't know what I was doing, but managed with a lot of help from other people and lots of Google, and so you can too. And then using these kind of tools like partial least squares to make inferences out of the data sources. I just wanted to give one quick example of something I do later in that paper. This is like figure 4 or 5 or something. And essentially, I had done this gene expression to term mapping. And what I wanted to do was also use this data set called Brainstem, which instead of being very spatially comprehensive, so that's to say it's not gene expression samples across the brain, it's for like 16 very specific brain regions. So it's way fewer brain regions. But what's the big perk is that they have this across development, like from like fetal stages. So like weeks post-conception until birth, until like adolescence, etc. Yeah, okay. And then I wanted to see, how do my gene expression patterns that I'm observing in this adult data, how does that change across development? And again, don't worry about the science. But the point here was that I need to use another data set, right? Like I've already been using 2, plus partial least squares analysis. That's not a data set, as a tool, but partial least squares analysis plus on-human brain, bless you, on-human brain atlas and Neurosynth. And then now I'm like, oh, let me do this other thing with Brainstem. It's a third data set that I'm trying to integrate into my project here. And if you go to Brainstem and there's a download button, you can click download, and it's a CSV. Like, it's actually surprisingly simple. It tells you the genes, it tells you the brain regions, it tells you the age of the person, it gives you the expression values. But I didn't do that. I knew somebody that already worked with this data set, and I asked them, like, hey, you know this thing well, I don't know it well. Like, can you help me out here? And you've already you've done the data cleaning already, you've like processed and gone through it. Could you with me and like tell me about your experience and help me out with interpreting this, because you're already kind of an expert and I'm like totally new. And like, this is something that I do very often, because in my work, because it's so, I'm integrating so many data sources from so many different places. In many ways, I'm an expert of like the method and not so much an expert of any of the individual data sources, right? And so to use these, I go to the expert, like the person who has worked with it before, or even the person who developed it in some cases. I mean, sometimes they're like really busy, but in other cases, like if they're happy to work with, like right now, for example, just as an aside, I was working with this and am working with this like Brainstem data set. I've never looked into Brainstem in my life. And I have learned so much from the expert on Brainstem imaging, who's like really awesome, nice, by the way. And like that wouldn't have happened if I hadn't reached out and said, I want to work with this data, but I don't like, I'm kind of new here and like I'm sort of green in the field. Will you like help me out and can we do something together? And she was like super enthusiastic about it. So yeah, secret to working with big open data is to like ask somebody to collaborate with you. Just ask the expert, you know, helps to be an extrovert. Get a PI who will ask for you. That's like the real, yeah. Okay, so that's like my very, very first paper. Again, I do want to emphasize that this is not something that I did when I was already an expert on doing these things. This is like when I first started. So I really like truly believe that anyone could do it. I was not a whiz kid, you know what I mean? Like you could do it too. And integrate these different data sources, different methods to come up with something that makes sense or that you believe in, that you can like write about and talk about and explain. Okay, so now I just wanted to say, like, I wanted to speak a little bit about how I found and used closed data, which is like really just how to send an email. That's pretty much what the whole thing is. But I'll just, again, tell you a bit of a story. This is another project that I did a bit later. And the end game is that I put together this receptor atlas. But when I say put together, I don't mean that I like got a bunch of patients and scanned them for different receptor tracers and then like put together an atlas. What I did was I contacted people that had worked with PET tracers before. And I said, hey, you've worked with this before. I know that because you published on it. Do you have the group average for the healthy controls? Would you be down to share it with me? A, first question, would you be down to share it with me so I could do my project? And B, would you be okay if I put it up on GitHub? And so I started local and went less local. And then eventually I just like collected, I had to make the font smaller for the people because there's so many people. I put together this data set of, it's altogether 19 different neurotransmitter receptors and transporters across 9 different neurotransmitter systems, a whole bunch of people. Each map is group averaged because of ethical constraints to sharing like individual subject data, right? So instead of sharing the individuals, they shared the group average, which is what I wanted to look at anyway, so I was okay with that. And it made the whole open science aspect of it a bit easier. Not to discourage like going through the extra hurdles, but it just, yeah, some people, I guess. Anyways, I'm not going to go there. Okay, so these are the different data sets that I kind of collected. And like one thing I liked it when I talk about this, I just want to make sure that it's clear that like the people who shared this data, they weren't like sitting on their pile of gold and saying like no one can come, but like Justine seems nice, so I'll give it to her. You know, like no, it was like I just happened to ask. And I was like, hey, would you be down to share with me and for me to make it open? And they're like, yeah, take it. Like, great, please do, you know? And they were like super happy to have it be used because this is data that they have like worked so hard to develop, but they might not necessarily have the support or the knowledge or whatnot of how to make it open, right? Like it's just not really on their radar for most people. I mean, not for everybody, but it might not be on their radar. And so if you come in as like, I'm interested in making a tool, or I'm going to put this up on GitHub or whatever, would you be okay with that? They're usually, or in my experience, they were very happy to share this data. And then at the end, that data gets used a lot more now. And so they're getting a lot more recognition because people are studying their work. There's the ones that like put it out and they're like, everyone's happy, everyone wins, right? Like you get to use the data, they get recognition for the data that they put out. So it was like overall a really positive experience. And how I contacted the people was really just sending an email. Hey guys, like I'm thinking of doing this project. Would you be willing? You have this specific thing. Would you be willing to share it with me? This is where the data sits now. So it's up on GitHub. It's also in a toolbox called Neuromaps. So by the way, this receptor thingy and the Neuromaps toolbox, this is what ended up with me winning that Open Science Award at the beginning that kind of kickstarted all this. So it wasn't like, I like did not know much about open science. I wasn't like, oh, I'm really a proponent of open science, so I want to do this. It was more like, I'm really interested in the mapping between receptor architecture and brain function. How are they related? I don't have the data, so I need to kind of collect some. And why not make it open? Because anyways, I was in the habit of using, like putting everything code and data up on GitHub. The reason I was in that habit was that Ross was in the lab and he was into open science. And I was like, you know, Ross is cool, be like Ross, like that whole thing. So I was in the habit already of putting code and data up on GitHub, so I let the collaborators know, hey, like, would you be okay if at the end I put this up on GitHub? They were like, yeah, that's fine. And then I did that, and then it kind of blew up because everyone was like, amazing, a receptor Ellis. Like, I would love to use this in my modeling study, or I would love to get access to the very specific serotonin, 4 receptor for my hypothesis about depression or whatever. And so it was used a lot, and then we ended up integrating into it a toolbox called Neuromaps, which actually does a lot of other cool stuff I'm not going to get into, but it sort of became this like open science thing that I saw the ad for the Open Science Award. So I applied to it, and I was like, well, I did some open science stuff, maybe I can like apply. And then I won the thing, and I was like, and then people thought I was an open science expert. So now I have to be one because of what people think, you know what I mean? So that's kind of how it works. It's kind of worked out though. Okay, so the advice. When you're asking about data that is closed, be specific about what it is you want. Because if you're really vague, they're just not going to answer your email. That's honestly what's going to happen. Know what you want and why you want it. I really recommend, I think I'm really fortunate that in my lab, we're very open with having more authors on the author list. I know for some labs, for some PIs, for some groups, it's like a little bit of a thing that you want to keep the author list short. Like, I don't know. I don't think that's healthy, I guess, in science. I don't want to throw out too many hot takes, but I think it's like, in general, collaboration is healthy. And they're the ones that collected the data. What I always say is, would you be willing to share this? You would, of course, be included as a co-author on the ensuing publication. I do suggest that. I think it's the right thing to do and the nice thing to do. Really importantly is to credit the original person who developed this data. So like, for example, I put this data up here, but I'm not the one that developed it. So I don't want people to just cite me when they use it. Even if that's maybe where they found it or that's where they got it from. They heard about my paper, they saw it on my GitHub repost, so they're citing me. Like, no, you should be citing the original person who put out this data. And that's why there's a little bit of effort that's needed to make sure that people know who that is and like how to cite. And, for example, I'm here telling you about this. Like, if you were to use this data, by the way, please cite the original author. I think this was really helpful in my experience, was proposing an initial meeting and having some slides ready. So at this point in my project, like at the beginning when I was trying to put together some data, I had a couple put together already from just open like NeuroVolt or open sources or data sources sort of in the cloud elsewhere. And so I had like a pipeline for my analyses, and I had some kind of preliminary results that were going to change a lot actually when I started adding a lot more data, but I had some slides essentially. So I could contact these people, say, I'm interested in this thing. Would you be willing to share it with me? This is what I'm planning to do with it. You would be, of course, included as a co-author. And would you like to meet? I can show you what I'm planning to do with it. And that way you're keeping things transparent and open. And sometimes they were like, no, we don't need to meet, like, go. But a lot of the times it was more like, yeah, for sure. Like, tell me what you're interested in doing with it so that I know how to like take it further. And then really importantly, like in all of these meetings at the very beginning, I tried to make it really clear. And myself and all this, my PA is very supportive. So he's definitely like a big support in all of this, and trying to doing open science when you don't have a supportive PI is like very difficult. And I like commend you if you're trying to do that, and good for you. I'm lucky that I have a PI who's very supportive about this open science stuff. In any case, so both of us would make it clear in these meetings, like we would like to eventually make this open. So it's clear from the beginning, because what's a really sticky situation is if you take someone else's data and then you don't have the permission to make it open unless they've explicitly given it to you. So you can't just, put it up on GitHub without making sure that they totally understand what is being put up on GitHub. So for example, in my case, it was, I would be putting that group average map up on GitHub, and then they would say something like, I'm sure that's okay, like no subject specific data. I was like, no subject specific data. In fact, I don't even have the subject specific data, it's just the group averages. So yeah, I think this is like really the key take homes for trying to get closed data and then eventually making it open. I totally recommend that if you are using closed data, do try to then make it open. It helps everybody. I think sometimes people don't totally know how much it helps them, like the people who develop the data, but it is actually like people then hear about them more, people are using the data more. I think it's good for science. I recommend it. Okay, this is my very last slide, or like my second last slide before the thank you slide. This is literally the entire slide. So credit where credit is due. I already kind of talked about that a lot, but I just really want to emphasize, like, I've never collected data in my life, which makes me a data leech. And as a data leech, like, it is absolutely our responsibility and duty to make sure that people know where we are getting this data from, who collected it. That counts as, do I have a slide? I don't think I do. Nope. Citations, co-authorships, When you make your open GitHub repo where you put your data, saying like, this is the person to cite. If you use this thing, cite this, or I use this thing, this is where I got it from. So this is where you can go to find it. Yeah, just it is important to keep in mind. Okay, so this is like my very general how I came into the whole big data, open data, integrating data sort of science lifestyle. I'm happy to talk about like literally anything else. If there's specific methods you want me to go into in more detail, if you want to know how I was accessing this or that, or how to, one big question is like, how do I like do this with my PI's approval? Like, what if you want to do all these like really big open science thing and your PI is kind of not really into the whole open science thing? How do you like get your PI on board? Because obviously you guys are doing this together. Okay, so thank you to you for listening. Toasty Open Science Office Hours, well, Toasty in general for the invite. My lab, I'm very lucky, they're very supportive. Data collectors, data sharers, like I'm not gonna like miss that after like preaching a bunch. And yeah, let me know if you guys have questions or anything you want me to talk about in more detail.